HIV or human immunodeficiency virus. HIV spreads through exchange of body fluids. Main causes of these transmissions may include unprotected sex or sharing of contaminated needles. Once inside the body, HIV infects cells that are part of the immune system, particularly targeting helper T cells, which helps defend the body against bacterial and fungal infections.
HIV is a retrovirus, which is a type of RNA virus that inserts a copy of its genome into the DNA of a host cell that it invades, co-opting them to make more copies of itself. So are there any treatments for HIV? There are different types of antiretroviral therapy. Some works by blocking HIV's access into the immune cells. Others work by stopping the virus itself from replicating. These drugs are highly effective at managing levels of HIV and preventing T cell counts from getting low enough for the disease to progress into AIDS.
These drugs are now increasingly effective. As a result, in the UK, the mortality rate has decreased from 1,723 deaths in 1995 to only 490 in 2012. These drugs, however, have unpleasant side effects, including headache, diarrhea, and even permanent nerve damage. In sub-Saharan Africa, which accounts for over 70% of HIV patients worldwide, antiretroviral treatments only reach 1 in 3 HIV-positive patients. Getting those medications depends on the access to a functioning healthcare system. So in order to stop the spread of HIV, it requires a huge amount of investment in order to improve those systems.
The first ever person cured from HIV was known as the Berlin patient. He had a stem cell transplant from a donor who had mutations in the helper T cell. Normally, HIV binds with the CCR5 receptor on the membrane of helper T cell in order to enter the cell and infect it. Therefore, a mutated CCR5 receptor means that the virus will no longer be able to bind and get into the cell.
This seems like a cure, but the risks, difficulty, and expense of this procedure make this a highly impractical approach to curing HIV in most people.
A different experimental approach involves gene editing known as CRISPR-Cas9. Cas9 protein could be programmed to search for a specific sequence of the DNA and cut the HIV out of the cell and let the DNA heal itself. But concerns about safety, efficiency, and practicality of this approach have so far limited its use to laboratory experiments. Although numerous methods have been tested and examined over the past 2 decades, no approaches have been powerful, safe, or effective enough to be successful yet.
So, when will there be a cure for HIV? Well, scientists are working day in and day out to find a possible cure. A cure might as well be tomorrow, who knows? And while 1 case out of 70 million cases cured may seem like terrible odds, 1 is immeasurably better than 0. We now know that a cure is possible, and that may be what we need to beat HIV for good. Thank you for watching. My name is Patra-Yanan.